I just want, my objective is just to give you an overview of testing. I will not go into much details because those of you who are interested in the details can go to the original publications and look at how these things were done and why. I just want to give you, again, kind of an overview of this issue of testing. And I wanted to start with this. This has been developed mainly by Ignacio Blanco, who is here with us during this meeting, and to give an overview of the situation of Alpha-1 around the world. And in fact, as you know very well, this is not so infrequent, particularly in some areas in the world, but it's still undiagnosed. And this is undiagnosed despite that more than 20 years ago, this WHO document suggested or recommended that we had to test. We had to test particularly respiratory patients, not only respiratory, but mainly respiratory patients with COPD, with emphysema, adults with non-reversible asthma. But in the last more than 20 years, we were not successful to test all of these patients. This was a paper that was produced by a collaboration in different labs in Europe, and we again reproduce this wish that we should test at least all patients with emphysema and all patients with COPD. But what is the real situation? And this is in Italy and Germany. And in fact, there are only a few doctors that test all patients with COPD. And I would say that I was positively surprised that in Germany, 97% of doctors in hospitals tested for Alpha-1. But in fact, in the offices, only 41 and 21 in Italy. And most of the patients with COPD, fortunately, are not admitted to hospital. They more often visit their doctors in the offices. So still, we are far away from that. But in other countries, and with the collaboration also with Maria Susena from Portugal and other colleagues from Spain, we copied that survey, and we did it in our physicians, and the situation was not better. In fact, all COPDs were tested only in pulmonologists, approximately 30%, and even less in internal medicine and primary care. And one of the reasons is the lack of knowledge. And in fact, if you look at these columns here, you may not be able to see the numbers, but you can see the size of the columns. Doctors who really said that they knew a lot about Alpha-1 were less than 50% in the case of respiratory specialists, and very few internal medicine and primary care. So in fact, we need to improve this knowledge. And it was not really different, again, from Germany and Italy. And you can see how doctors that declare to know very much about Alpha-1 in Germany were approximately 53% pulmonary specialists, Italy 26%, but very few in primary care or internal medicine. And again, if we ask those physicians how many of them tested all COPD patients, you see that only 18% or 25% in Germany and Italy, respectively, declare that they test all COPD patients. So this is a very low percentage. Obviously, they think about Alpha-1 in young COPD individuals or family members. That's very clear. But, I mean, you know very well that not only those who are younger than 45 should be tested, but all COPD patients should be tested for the deficiency. And there was a variety of reasons. One of the reasons was the lack of knowledge, and also the lack of knowledge was represented in this slide. Some said, well, there is no treatment available, it takes much time, the cost for testing is too high, or even some say, I don't see those patients. Well, if they don't test, obviously they don't see them because they don't recognize them. Yeah. And when we look at a large database in Catalonia, primary care, more than 6 million people, 80% of the population, and we look at the testing for Alpha-1, and these are the testing for different years. And you see in different years it went from approximately 2,000 to 3,400, 600 per year. Well, to put that in context, in the same database we have approximately 8,000 new COPD patients every year, so new diagnosis, on top of the already existing COPD patients, or the liver disease patients, children, and so on. So this is clearly a very low number of tests, and I don't have it in this slide, but if you see the publication, in some of these cases, the reason for testing was not clear. It was not that all of these individuals tested had a disease susceptible to be caused by Alpha-1. So the reasons was really unclear. So there were a very few and not really focused. So at the end, and you can see the distribution of ages here clearly, a peak in children and then in adulthood. But it, again, demonstrates that we still need a lot to do. And this is probably not surprising, and because these are data from the Spanish registry with Beatriz Lara and myself, and you see that only 3 doctors in Spain saw more than 40 patients with Alpha-1. But the majority of doctors only see 1 or 2 patients in their life. So this is, again, stressing the need to have reference centers, to have registries, to accumulate the experience, and therefore to give guidance to other non-specialized physicians about the Alpha-1. Because it's difficult that doctors that see only 1 or 2 patients in their lives can accumulate enough experience with the disease. Well, back to the European paper, we also highlighted the advantages and disadvantages of the different samples that we can use for testing. And the main samples are whole serum or plasma or dry blood spots. Again, I will not go into details. I will not explain all these slides, so probably you are quite familiar with these different ways of testing. All of them have these pros and cons. There is not a perfect sample. So for screening programs, dry blood spots are better because they can be handled, they can be shipped. But for full analysis, full testing, genetic testing, and so on, sometimes blood samples, whole blood samples are necessary. And there is not even a unique algorithm for testing. And in that paper, we describe the different algorithms from Italy, Germany, and 1 lab in Spain, our lab in Barcelona. And in Italy, they start with dry blood spots, and they do measurements by nephelometry and genotyping. And then if they are consistent, that stop, or then they follow to phenotyping. In Germany, they start with the testing by individual physicians, and then when the levels were low, then they again collected dry blood spots. We in Barcelona, we started with dry blood spots. If the concentrations were normal, we rule out the deficiency. Otherwise, we continue all the protocol with genotyping, phenotyping, and so on. At the end of the day, there is not a good or a bad algorithm. They all provide the same diagnostic yield. It's just depending on what techniques do you have available in your lab and which is your expertise. To make it a bit more simple, in our Spanish recommendations for Alpha-1, we recommend that in general, not in specialized labs, but in general, that first you have to check serum AAT concentrations in stable clinical conditions. We have talked a lot about the acute phase reactant nature or category of Alpha-1, so it's important to do it in a stable clinical condition. If it's normal, it's normal. If it's low, then you have to go to phenotype. We prefer to go to phenotype in general, although some labs can do genotype first for C and S allele, and only genotype in those who are not the usual phenotypes. As I said before, it has been developed, the technique with dry blood spots. Dry blood spots are very useful and are useful for doctors that do not have a reference lab near available, so they can ship the samples. And we developed this some years ago, but other labs have been doing this for many years, like in Florida and in other places. And with this technology, we developed a screening program, a case detection program in COPD. That was, well, relatively successful. We collected a lot of samples and we could identify different individuals with different phenotypes. But then we tried to extend that in primary care. And actually it worked in primary care. The problem is that at the end of the day, you see, this is the ratio of patients per doctor participating. And it went up to a mean of approximately between 6 to 7. But these doctors see a lot of patients with COPD. And they tested only approximately 6 to 7 per physician. So they were really not motivated to do that. Even if you have the best testing technology, if they don't think about it and they are not motivated to do it, then you are not going to succeed. And this is really one of our hurdles that we have now. We even perform a cost-effective or a cost per diagnosis analysis and in all of our program. And the cost per PICC detected was approximately 3,600 euros, which is quite reasonable for all this setting that we have to develop. But if this is very sensitive to the different technologies you use, how you ship the samples, so you can do it even cheaper. I mean, but it's quite reasonable. Other countries have developed also these testing strategies. This is published in Germany. And they were very successful. They collected thousands and thousands of samples, and they had a very reasonable yield, 7% of CC alleles, and some heterozygous, deficient heterozygous, which is quite good. When looking at the clinical diagnosis of these patients, I was surprised to see that bronchiectasis was in the first place, probably because these patients are seen in specialized clinics, and therefore those physicians are more motivated to do a series of testings compared with emphysema or COPD that are attended, but with a lot of physicians that are not particularly specialized either on COPD or in respiratory medicine. Another experiences in Poland, in this case, they also collected an important number of samples, but the diagnostic yield here was smaller, only 1.5% of CC patients detected. So it depends on the population you select, obviously. And also looking at the geographical distribution, you see that most cases were around the location of the reference lab. So, and there are areas of the country that almost did not participate in the screening. So it's important that all patients or candidates or individuals susceptible can have access to this diagnosis, to this testing. Another experience, just to highlight the importance of motivation in Germany, they distributed more than 17,000 kits for testing, but only 2,700 were submitted. So there were 15,000 that were lost and were not used. So that's, again, we need to, and I will insist on that, we need to motivate the physicians to really test the patients. And they developed different strategies, different congresses, lectures, and after each one of these activities, there was a peak in sending samples, but then quickly it returned to baseline. So the motivation is not long-lasting, actually. Well, we were also discussing about the different strategies to increase the awareness, and you can think about many different strategies. Some of them are more successful than others, but we are still struggling with this. And there are some innovative strategies. This is Marc Brownlee is here. So they use a video with short instructions, and then with this, they increase the number of the knowledge of the disease. And with this, we assume that we also may increase the number of testings. Even more, probably more effective could be that doctors, and in particularly primary care physicians, have some alerts in their computers, in their systems, when they see a patient with COPD, that they may have an alert saying, well, this patient has COPD, so have you tested for Alpha-1? You should test for Alpha-1 because you should test all patients with COPD for Alpha-1. And then when they have these warnings, by using these warnings, you may increase testing in those patients, as was demonstrated in this study. This is something that we are now also struggling to implement in the electronic medical records in primary care in our community. And finally, we have different strategies also to increase the testing, and this is the Alpha-Kit quick screen that can be done in the doctor's office in a few minutes, and it will tell you if there is any C allele present. It's not a definitive test, but at least it helps you to screen populations without the need to ship samples initially. Other strategies, and this is the end of my talk, that we are now developing is swabs, in which that means that you do not need to take blood samples for the individuals, and we have already published our validation of genotyping using these buccal swabs for the C allele, the S allele, and the M malton, which is very prevalent also in Spain. So in summary, in general, we first need to do a quantitative analysis of alpha-1 and consider CRP for these issues of the active phase reactant. Then phenotyping for common variants and or genotyping, depending on the availability in your lab, and only genotyping sequencing in those cases in which results are discordant. Don't forget that it's not just the patient. There is also family screening for parents, siblings, and partners. And remember the genetic counseling and the legislation in each individual country regarding genetic testing of individuals and of patients and relatives. And finally, and this is my last slide, this would be my wish, that every patient with COPD was tested at least once for alpha-1, and this should be our aim. Thank you.
First of all, I would really like to thank the organizing committee for inviting me. As a newcomer to the field, this is especially important to me, and I had a great time here, and I'm looking forward to continue the momentum. So I would like to, at the beginning, share a little story how I actually got into the field. So I have a long-term interest in protein inclusions, such as these Miravitlles bodies. And because of that, I was asked to write a big review summarizing what kind of inclusions exist in the liver. And by doing so, obviously, I bumped to all the inclusions that are found in endoplasmatic reticulum. And given that I have kind of some spare money in my pocket, I thought, well, this would be fun to look at it closer. And first contacted Jeff Techman to get the PIZ mouse and actually to get some nice pictures for the review as well. And that's how I got started. So in the process, I got to meet Sabina, where we discussed alternative animal models for alpha-1 antitrypsin, and Sabina was really a long-term mentor, and I would definitely not be here when it would not be for her. So I would like to thank her for that. So at some point I thought, well, mice are fun, but you know, you are a doctor, so you have to go to the physicians, and again, the person who kind of hooked me up into the community was again Sabina, and then I started to learn all the field, and I would like to really thank Marion, Gabi, and Frank, who are really the driving forces behind all these things, and again are very important for that I am here now. So I would like to talk briefly about our work we do in the PIZ mice, but then I will, in the second part, I will focus mainly on the patient-related issues. So I have a long-term expertise in transgenic animals, and as I said, I am very passionate about protein inclusions, and because of that, I was wondering how different, let's say, human-related protein inclusions may work together, and what happens when there are patients which may end up both hepatitis B, for example, and alpha-1 antitrypsin. So in hepatitis B, you get one of the viral proteins called hepatitis B surface protein gets accumulated in the ER and forms what's called ground glass inclusions. And what's not that maybe commonly known is that this is something what we cannot treat at all. So we can treat hepatitis B, and this brings down the level of the virus, but we cannot treat the hepatitis, the HBS protein production. So in that sense, we cannot really help so far in that sense. Although both protein aggregates are technically in the ER. They don't overlap at all, and we are trying to figure out what are these different accumulations pattern doing, and why is it like that. So as expected, the mice are doing pretty bad. They develop a stronger fibrosis, and later on they develop a lot of tumors. And in terms of the underlying signaling, there is the classic prototoxicity signaling, autophagy-related stuff, and so on. But in sake of time, I would like to cut this a little bit short. And P62, which is one of the classic autophagy deficiency markers, is really lining up quite nicely with the AAT. So there is a clear association. So as I said, at some point we decided to not only look at mice, but also to talk to the people, as we physicians do, and started a study where we basically combine a detailed questionnaire. We do a relatively extensive blood biosampling. A lot of people in the room can confirm that it's quite extensive. And then we use this elastography, which I think is really for Alpha-1 the way to go, because as every technique, it has some drawbacks, but you can really reliably exclude a significant liver disease in at least 50% of the patients, and on the other hand, you definitely quite reliably can recognize the severe ones. And obviously, there will be still people needing liver biopsies, but this should reduce the need for liver biopsies by at least 60–70%. So this is the stage where we were at probably 2–3 months ago. Now we've got a few more people. So we analyzed about now 350 ZZ patients. We have about 180 MZs, and totally, also with the controls, we are pushing towards 800 patients. So we are still our strong base is in Germany, but we did kind of invade Austria pretty successfully as well. We are collecting in Czech Republic, and with Gunhild, I guess in a few weeks or months at the latest, we will invade Denmark as well. So and actually in Netherlands, we are also just around the corner. So the results are probably not surprising to too many. So one important thing which is to stress out is, again, most of the Alpha-1 patients don't have elevated liver enzymes. So depending on what liver enzymes you look at, 20% or maybe even less patients have elevated liver enzymes, and the correlations with liver fibrosis, which is really the key issue. So the key issue, how sick is your liver, is the amount of liver scarring, not the amount of liver enzymes, because the liver enzymes tell you whether you slept well the last night, but it does not tell you what's happening to your liver for the last 20, 30, 40 years. So this is one of kind of important caveats. Obviously, if you have elevated liver enzymes, that should prompt you to go to a physician and get kind of your liver checked even more urgently. But if you don't, you should still do the same, basically. Good. This is just a brief summary of the FibroScan, so of the fibrosis data. So we see about 30% of patients having a significant liver fibrosis, which is like intermediate or advanced stage. What is probably even more striking is the data on steatosis. So if you use the established cutoff values, 70% of alphas have a liver steatosis, and we are trying to understand that more, but this is really the most kind of obvious issue, and it looks like that as an Alpha-1 patient, you are predisposed to steatosis, irrespectively of the normal risk factors such as obesity and alcohol intake and whatever. So that was just a brief overview of the ZZ patients and of the general study where we are at. And another thing which I was very passionate is about the MZ patients, because obviously there is quite some amount of literature suggesting that MZ patients may be at risk to develop liver disease, but the data is kind of not present in the field, and if you look at kind of hepatologist reviews, it actually never shows up. And this is part of the problems with genetic studies, that if you have 1 or 2 genetic studies, if they are not too big, people will not believe them until you get them confirmed over and over again. So that's what we did. And we kind of took the best stuff out there, which is usually the good thing to go. So we took basically the cohorts which were used to identify the genetics of alcoholic liver disease. So these patients are all heavy drinkers, and in heavy drinkers, part of them develop liver cirrhosis, part of them don't. So they were about 50-50, so 50% were cirrhotic, 50% were non-cirrhotic. And as an aside, in this very well-established cohort, we found actually 2 ZZs and 3 SCs, which kind of, despite genetic testing and everything perfect, nobody picked up. But what is probably more interesting is the odds. So if you are an MZ, you have about 5 times higher odds to develop liver cirrhosis when you, at the same time, kind of enjoy alcohol a little bit too much. And this is the same whether you look at adjusted odds ratio or the unadjusted odds ratio. And we are talking about p-values to the power of -7, so that's kind of not any question, basically. So we also looked at non-alcoholic fatty liver disease, which is kind of the modern liver disease and the hope for hepatologists' future. So this is disease in patients who are obese or inactive. Diabetics are very predisposed to get non-alcoholic fatty liver disease. So these were all patients who got liver biopsies. So although it's only 750 cases, it's a very clean cohort. And here also you already can see that the MZ patients are enriched in the ones which already have liver scarring. So what we see in kind of patients with advanced liver diseases, we see up to 10% of MZ patients. So it's also kind of a significant population. It's not just like a rarity or something like that. So it's a significant proportion of these patients. And again, when we do the math, we see a odds ratio of at least around 3. If we look at only the cirrhotic versus non-cirrhotic, we see similar odds ratios. Again, this cohort is a little bit too small to reach really impressive p-values, but at least most of the data are quite nicely significant. So that brings me to the conclusion of my talk. So I showed you a little bit what we do on the basic research level. I showed you even more what we try to do on the patient level, but we are still at the very beginning, and it's my pleasure also to announce that we got an EASL registry grant. That means that we were asked by the European Liver Association to develop a registry to collect all patients with Alpha-1 and to really systematically study them. And I hope that this is a great opportunity for us to really raise the awareness about the disease and to get some collaborative work done. So if anybody is around in Amsterdam in 2 weeks, we will be happy to see him and kind of join forces together. And last but not least, I would like to mention the guy who did most of the work. That's Karim Hamesh, a very hard-working physician in our department. Thank you.
First of all, I would like to thank the organizer committee to invite me to present my work about genetic mediators of Alpha-1 antitrypsin deficiency mediated liver toxicity. I also named this presentation The Bad, the Ugly, and the Good. You will understand later why. First, I would like to introduce myself. I am a researcher. I am a French researcher. I work in Bordeaux, just here, for the French institution, the CNRS, and INSERM. My work focuses, as you can guess, on alpha-1 antitrypsin, or AAT. AAT is produced by the liver and released in the bloodstream where it acts at the lung level. AAT is mandatory for maintaining lung integrity. When you develop alpha-1 antitrypsin deficiency, that manifests by emphysema and also liver damages. As a researcher, part of my work focuses on the liver. But I don't focus on all the organs. I focus on a specific cell in the liver named hepatocyte. You can consider these cells as a factory of AAT. In fact, I check inside these cells to understand what happened exactly in these cells, why the AAT is not released from these cells. You can imagine this cell as a square. AAT is produced and released in the bloodstream. But when AAT is mutated, deficient, it's like a policeman involved in the quality control will declare AAT as mutant and will address this protein as a trash. Consequently, AAT is not anymore released in the bloodstream, and you don't have any more AAT at the lung level, and you will develop emphysema. For some mutant, as a Z mutant, this mutant is also addressed to the trash, but this mutant have also a second property: it can aggregate. This aggregation are involved in the liver damages. But there is a specificity about liver damages, because about just 10% of the population homozygous for the Z variant develops a clinically significant liver disease. There is also considerable variability in the age of onset and severity for this liver disease. All this clue means that genetic modifier or environmental factor might contribute to the risk and severity of liver disease. If you want to sum up, you have the bad guy, the Z mutant. You are at risk to develop emphysema. But if you carry also the ugly guy, genetic modifier, you are at risk also to develop liver damages. Unfortunately, so far, we don't know which are these genetic modifier. It's for that a part of my goal, my research goal, is to identify which genes are involved in the alpha-1 antitrypsin-mediated liver toxicity in order to find the good, the therapeutic avenue to treat, to prevent all the symptoms associated to the alpha-1 antitrypsin deficiency. The strategies that I use to identify the genetic modifier is the whole exome sequencing strategy in family in which the ZZ patient with liver damages has a ZZ brother or sister free of any hepatic sign. But what is the whole exome sequencing? The whole exome sequencing is I will check every molecules, every letters of your DNA. It's for that, I need your DNA, but don't worry, it's not a problem because it's not painful. I just need saliva, for example, with a saliva DNA collection, or a little bit of blood. I just need some cells and extract your DNA. When I get your DNA, I will compare the DNA from the patient with hepatic injuries and his sibling without any liver dysfunction, in order to highlight genes mutated, genes different, exclusively related to the liver damages, exclusively present in the patient affected sibling, in order to highlight, to get candidate genes or a list of potential targets. But of course, all the genes are not relevant. It's for that I need a second step. I need a validation in experimental system as human cell lines, mice, or exotic experimental system as worms or yeast. every step is to identify the good, the new therapeutic way to prevent all these symptoms. Indeed, the identification and the validation of gene alterations that cause liver disease aggravation in Alpha-1 antitrypsin deficiency patients could consequently indicate a risk patient at an early stage. This patient will therefore be subject to a more intense clinical surveillance and perhaps placed on the transplant list at an earlier time. But okay, so far, what I got has results. I already sequenced 5 families from the Defi Alpha cohort in France in collaboration with a medical group in Lyon. And from this sequencing, and after a bioanalysis of this sequencing in collaboration with a group in US, in Stanford, we found 2 candidate genes. Using these 2 candidate genes, we validate them in different experimental models, as I already showed you. And what we got is we realized that the policeman is involved in the liver damages. In fact, the policeman doesn't work correctly, so will induce the aggregation and increase in the Z accumulation, and consequently the death of the cells. Our findings provide promise for early prognostic for Alpha-1 antitrypsin deficiency associated liver disease and could even lead to possible pharmacologic development that is for the good. But as you can already notice, we sequenced just 5 patients, 5 families, 10 patients. It's for that our conclusion needs to be confirmed in a larger group of affected ZZ patients before the definitive role of these 2 candidate genes can be sustained. And it's for that I need you. I need your DNA. Okay, I would like to thank all the persons involved in this project, in particular the medical group in Lyon, also the bioinformatics group in US. I would like also to thank all our funding as INSERM, La Ligue, le CNRS. And also I would like to give a very, very special thanks to the French Alpha-1 Antitrypsin Deficiency Association. This association fights against the disease and supports me a lot in my work. And as I say already, every day I know that I'm not alone in this fight. The people of the association are with me and support me. Thank you very much for your attention.
And we have now a few minutes left for questions and answers from the panel. You have been very good in holding back your questions. I'm inviting you to ask them. Well, okay, excellent. One question from there.
Hearing from all 3 of you, I would like to make a modest suggestion that the 3 of you travel throughout Europe. We have Pavel. Pavel, you're already going to many countries, and you should be teaming up with Marion, so while you're doing the FibroScan, she's taking blood samples. And then, Marc, you would be talking to the GPs to get them excited about identifying their patients. I'd like you to talk about that team.
Any comments?
You already talked about that with Pavel, and we try to collaborate together now. Yeah, we try to set up this collaboration.
There was a question here in the front.
Thank you all for this presentation. I feel like I'm watching an episode of CSI. And the cost of a swab and the analysis, as you've shown us, Marion, and some of the other screening techniques make it feasible for us to disseminate this research, essentially, or the discovery. Are there programs that are in Europe now where you're promoting the screenings of this at the local level without having to work your way through the entire physician establishment or the medical establishment, but instead go right to the people where they can then say, I need this help. It costs me a very little bit of money, and this is going to help me and my country and my people get better.
Marion, Pavel?
Could you repeat the question? I didn't check.
Well, first, it seems inexpensive, relatively speaking. So to get the data, and the people will make the decision. So can you comment upon the expansion of the detection systems, and then the action will come from groups like this, where we're organized to try to bring that knowledge to the people of the government, the healthcare reimbursement system, all the above.
Yeah, to collect the DNA is not expensive. The most expensive is the sequencing. The sequencing costs for one person 1,000 euros.
Well, for us, we do all these things for free for the patient. So I think that the issue always is an access to patients. So for us, we analyzed, let's say, in Germany, roughly the numbers as are the members of the Alpha-1 Deutschland Patient Association. And of course, it's not trivial to get the people because they are just spread out, and we do so we have Frank as our TV star, and we have some spots once in a while, and all these things. So raising awareness is kind of the issue. So it's not about us asking money for anything. It's more about the people getting to us and knowing about us. Also, in our case, still to have a good network of physicians, ideally throughout Europe, that hopefully in 2-3 years we will have 30-40 physicians that whoever calls or emails, we can say somebody around, 200 kilometers away is there, and you just can go there and get your test for free. That's the dream. Let's put it this way.
I have a question for Marc. If we had newborn testing, we wouldn't have to worry about our testing failures. And if newborn testing for Alpha-1 was included in the screen, we wouldn't have to discuss any of this. Marc, what is your opinion on that?
This is a very controversial issue because it has been done. It has been done in the US. It has been done in Sweden. It is very well known. And in fact, obviously, then you have the results. You know the person from the beginning, if the person is affected or not, and you may take preventive measures. And in fact, following this newborn cohort in Sweden, they realized that there were less smokers compared to people in other generations that were not submitted to screening, and also compared to those who were not identified as deficient. So that's a good thing. But in contrast to that, there was also an increased stress in the parents, and in particular in the mothers. And in some cases, it was not justified. So it also created some side effects. So, and therefore, it has not been widely recommended by scientific societies to perform newborn testing. But this is always on the table. I mean, and there has been debates and debates about the pros and cons to newborn screening. But so far, based on those experiences, it's not widely recommended, and we do not do it in Spain.
I tend to agree with respect to adult manifestations of alpha-1 antitrypsin deficiency in adults. But most newborn screening is done for pediatric diseases to prevent pediatric manifestations of genetic disorders. So I was just wondering what the hepatologist's opinion is on this, realizing that liver disease is the predominant manifestation of alpha-1 antitrypsin deficiency in children.
Well, I mean, even for liver, about single-digit percentage of Alpha-1 patients develop a severe disease. So I mean, there is a clear recommendation to test people, newborns with prolonged jaundice, and so there are kind of the phenotyping-based recommendations are out there, and I kind of agree that, of course, testing everybody and getting 2% of population in Europe is MZ, and then if you don't have people trained for genetic counseling and so on, and then you need 1 in 50 patients, you have to, or mothers explain that they have a heterozygote mutation, which is something which is difficult to handle. I think it's important to do careful phenotyping and careful or like a proactive testing of patients who are resembling something like Alpha-1, but I think newborn testing may really cause quite a bit of harm as well.
In the United States, there are 3 criteria that are being used in most states. In the United States, each state determines what diseases are included in the newborn testing panel. And the recommendations to the states coming from the federal authorities is that testing should meet 3 criteria. Number 1, it has to be feasible and simple. So you can just do a simple blood spot to make the testing, do the testing. Number 2, it has to be affordable and cheap, because everybody will be tested. So it's a cost factor as well. And the third one is there has to be a prevention or a preventive intervention that is proven to be effective to prevent the disease from occurring once you have identified that somebody is at risk. And unfortunately, as far as we know, in alpha-1 antitrypsin deficiency, you cannot prevent liver disease in alphas with any kind of intervention. And so the only prevention we know for adult disease, which is smoking-related COPD, is a prevention of smoking initiation in children. And there is some effect there. But I guess, again, since newborn testing is usually focusing on children, that's kind of a very weak argument to use. Any other questions? We are almost on time, Honi. So I would like to...
Adam, may I make a comment?
I'm sorry, yeah.
I wanted to recommend that since you've connected the issue to smoking cessation, that one of the best ways to promote smoking cessation is to create a law at the local level, not the federal or the provincial or state level, but at the city level. And our city in Calabasas, California, has created a law that bans secondhand smoke, not tobacco, but secondhand smoke. And I would be happy to make that available to everyone. They can take it back to their mayor or their city councilor, and they can be installed. And in the process, then you localize this, and you make people aware of all the things that you all talked about today, and how to deal with it at the local level, and do it with a simple law. You don't have to spend the 60,000 euros to get the law written. It's already been done, and we'll be happy to share.
Thank you for that comment. I would like to thank Marion, Pavel, and Marc for their presentations. They were very thought-provoking, and you're now excused.